Currently there is no evidence about life on Mars, but during the Viking mission scientists did found some evidence of metabolic activity on the Martian surface, but in another experiment no trace of organic material was found. With no organic substances, what could be, or seem to be, metabolizing? So the common conclusion was that there is no life on Mars, but then why the results came positive in the LR experiment conducted by Viking 1? Well, before launching the Viking spacecraft, the researchers tested the experimental protocol on a wide variety of terrestrial soils from harsh environments, from Death Valley to Antarctica. In each case, the experiments tested positive for life. Then as a control, the researchers heated the samples to 160 degrees Celsius to kill all life forms, and then retested. In each case, the experiments now tested negative. To further confirm that the experimental procedure would not produce false positives, the researchers tested it on soils known to be sterile, such as those from the Moon and the Surtsey volcanic island near Iceland, which produced negative results as expected. Then why positive on Mars? The Viking 1 lander's LR results show that, when injected with the nutrient solution, the soil sample exhibited strong radioactivity, indicating metabolism. The control soil sample, which had been heated to kill any microorganisms, had a negative response. Later, Mars Science Laboratory Curiosity discovered organic molecules on Mars. Then why was Viking not able to detect them? Well, the answer to that could be that Viking's gas chronograph mass spectrometer was not a life-detecting instrument and required 1 million microbial cells to detect organic matter. Other researchers who support this view have proposed that Martian life could take the form of methanogens, microorganisms that produce methane as a byproduct, halophils, which can tolerate high salt concentrations as well as severe radiation and low oxygen concentrations, or some type of cryptobiotic microorganism that lies dormant until reactivated, such as by a nutrient solution like the one in the LR experiment. Note, to rule out the possibility that the strong ultraviolet radiation on Mars might be causing the positive results, the landers collected soil buried underneath a rock, which again tested positive. The control tests also worked, with the 160 degrees Celsius sterilization control yielding negative results. In addition, it seemed that whatever was doing the metabolizing was relatively fragile, since metabolic activity was significantly reduced when heating the sample to 50 degrees Celsius, and completely absent when storing the soil in the dark for 2 months at 10 degrees Celsius. Levin and Straught believe that these results provide some of the strongest evidence that the soil contained Martian life. There are certain microorganisms who can survive in harsh climatic conditions, so the possibility of life on Mars cannot be ruled out.